is about the genetics of axis specification in Drosophila. Let me try to learn about the steps of pattern formation in Drosophila. First, there will be symmetrical precursor cell, then formation of asymmetries imposed by cytoskeleton, then binary decision, positional information established, chemical gradients, soma and germline, primary gene expression domains established, broad domains of cardinal genes, then refinement cell-to-cell signaling, fine-grained expression domains, then repeating units or segments, dorsal ventral cell layers, positive feedback loops. Okay. Then early development in Drosophila. When we try to learn about this topic, we need to know something. So after fertilization, the eggs of the embryo specified by syncytial specification were the morphogenetic determinants diffused throughout the whole embryo that then direct differential gene expression in each cell. The initiation of anterior, posterior and dorsal ventral axis difference is controlled by the position of the egg within the mother's ovary and the interaction between the egg and its surrounding follicle cells. So insects egg undergo superficial cleavage produces initiation, a single cell with many nuclei residing in a common cytoplasm. When the nuclei reach the periphery of the egg during the 10th cleavage cycle, each nucleus becomes surrounded by microtubules and microfilaments. The nuclei and their associated cytoplasmic islands are called energits. After 30th division, oocyte plasma membrane folds inward between the nuclei after partitioning of each somatic nucleus into the single cell. This process creates the cellular blastodon, in which all the cells are arranged in a single layered jacket around the yolky core of this egg. At cycle 4th of the zygote, gene transcription is greatly enhanced and slowdown of nuclear division, cellularization and concomitant increase in new RNA transcription is referred as mid-blastular transition. Then primary axis formation during oogenesis. Each oocyte is descended from a single female germ cell, the oogonium, which is surrounded by an epithelium of the follicle cells. The oocyte is present at the posterior end of the egg chamber, as oocyte will develop numerous mRNAs made in the nurse cells and are transported on microtubule through the cellular interconnections into the enlarging oocyte. Anterior and posterior polarity in the oocyte. The Gherkin message appears to be synthesized in the nurse cells, but it becomes transported specifically to the oocyte nucleus. Inside the oocyte, it becomes translated into the Gherkin protein, and this time the oocyte nucleus of oocyte is very near the posterior tip of an egg chamber, and the Gherkin cell signal is received by the follicle cells at position through a receptor protein encoded by torpedo gene. This signal results in the posteriorization of these cells. The posterior follicular cells recruits the part 1 protein to the posterior edge of the oocyte cytoplasm. Part 1 protein organizes microtubules specifically with their minus and plus ends at the anterior and posterior ends of the oocyte respectively. The motor protein kinesin is an ATPase that uses the energy of ATP to transport mineral to the plus end of the microtubule. The message transported by kinesin along the microtubule to the posterior end of the oocyte is OSCAR mRNA. OSCAR protein recruits more PAR1 protein, thereby stabilizing microtubular orientation. Then OSCAR can bind the nanose message. Dynein is a minus end directed motor protein that transports its cargo the opposite way. The bicoid message binds to the dynein. Dynein moves the bicoid message to the anterior end of the egg. The cytoskeleton rearrangement in the oocyte is accompanied by an increase in oocyte volume owing to transfer of cytoplasmic components from the nurse cells. This includes maternal messengers such as bicoid and nanose mRNAs. These mRNAs are carried by motor proteins along the microtubule to the anterior and posterior ends of the oocyte respectively. The protein products encoded by bicoid and nanos are critical in establishing anterior and posterior polarity of the embryo. Then establishment of AP axis in oogenesis and bicoid nucleization by Gherkin signaling. In early stage egg chambers, mTOQ is in the oocyte encoded mRNA is localized at posterior. So translation and limited diffusion means signals sent to overlaying posterior follicle cells receiving via torpedoreceptor. A signal is sent back which activates protein kinase A in the egg. Oocyte cytoskeleton is reorganized and directs the internalization of bicoid and OSCA, defining the AP axis. Then segmentation and the anterior posterior body plan. The hierarchy of genes that establish anterior posterior polarity and divide the embryo into specific number of segments with the different identities. This hierarchy is initiated by maternal effect genes that produce mRNAs that are placed in different regions of the egg, results in activation or suppression of certain zygotic genes. The first zygotic genes are called gap genes that are activated by maternal genes protect the defined broad territories of the embryo. The gap genes enable the expression of the pair rule genes, each of which divide the embryo into regions about 2 segment wide. The protein encoded by the pair ruled genes are TFs that activate the segment polarity genes, which divide the embryo into segment sized units along the anterior posterior axis. At the same time, the protein products of gap, segment polarity, and pair ruled genes interact to regulate other class of genes, the homeotic selector genes, whose transcription determine the developmental fate of each segment. Model of Drosophila anterior posterior pattern formation. Maternal effect genes, zygotic genes, syncytial blastodon, cellular blastodon. Bicoid as the anterior morphogen and nanos as the posterior morphogen. Bicoid mRNA is stabilized in the most anterior portion of the egg, while nanos mRNA tethered to the posterior end of the unfertilized egg. After fertilization, bicoid protein highest in the anterior end and nanos protein highest in the posterior end. Then the nuclei is formed. These protein gradients activate transcription of genes, specify the segment identity. So anterior group, bicoid repressed caudal mRNA, exuberantia anchors bicoid mRNA, shallow anchors bicoid mRNA. In case of posterior group, nanos repress hunchback mRNA, OSCAR localization of nanos protein. Pumilio helps nanos protein to bind with hunchback message. Caudal activate posterior terminal genes. And in case of terminal group, torsomorphogen for termini. Table of maternal effect genes that establish the anterior posterior axis of Drosophila embryo. Normal development, wild type bicoid mRNA, wild type phenotype. Then bicoid deficient phenotype, 2 tails. Normal development, head in middle, 2 heads. If we had normal development, wild type, head, tail, wild type phenotype. But development of bicoid deficient mutant, bicoid deficient phenotype, 2 tails. Okay, tail on both the ends. Then next experiment, add bicoid mRNA to embryos. Add to anterior end of mutant, head, tail, normal development. Then add to middle of mutant, head in middle. Then add to posterior of wild type embryo, wild type, 2 heads. An experimental demonstration that the bicoid gene encodes morphogen responsible for head structure. The phenotype of bicoid deficient and wild type embryos are shown. So when bicoid deficient embryo are injected with the bicoid mRNA, the point of the injection forms the head structures. And the posterior pole of an early cleavage embryo is injected with the bicoid mRNA, head structure formed at both the ends or poles. Summarizing early anterior posterior axis specification in Drosophila, genes that define the anterior organizing center. Bicoid protein gradient found in anterior end of embryo. Bicoid functions as TFs to activate anterior specific gap genes and as a transcriptional repressor to suppress posterior specific genes. Genes that define posterior organizing center. Nanos protein gradient found in the posterior end of the embryo. The center acts translationally through the nanos protein to inhibit anterior formation and transcriptionally through the caudal protein to activate those genes that form the abdomen. Genes that define these terminal boundary regions. The boundaries of afferon and telson are defined by the product of the torsogenes, which is activated at the tip of the embryo. Then dorsal ventral patterning in the oocyte. Gurkhan protein diffuse only a short distance, so the protein reaches only those follicle cells close to the oocyte nucleus, and it signals those cells to become the more columnar dorsal follicle cells. The Gurkhan torpedo signal that specifies dorsalized follicle cells initiate a cascade of gene activities that create the dorsal ventral axis of the embryo. The activated torpedo receptor on the presumptive dorsal follicular cells inhibit the expression of the pipe gene. On the presumptive ventral end, in the absence of Gurkhan protein, inactive torpedo effector facilitates the expression of pipe gene. Pipe protein modifies an unknown protein and allows it to be secreted from the ventral follicle cells. Pipe activates the noodle protein, which is secreted to the cell membrane of the neighboring ventral embryonic cells. Noodle protein interacts with its specified factors to split the product of the gastrulation defective gene. The GD protease cleaves the snake protein, activating snake protease, which in turn cleaves the Easter protein. Dorsal ventral patterning in the oocyte. As the oocyte volume increases, the oocyte nucleus moves to an anterior dorsal position. So the Gurkhan is active only in oocyte and torpedo. Its receptor is active only in the somatic follicle cells. Gurkhan protein diffuses only a short distance, so the protein reaches only those follicle cells closest to oocyte nucleus, and it signals those cells to become the more columnar dorsal follicle cells. The Gurkhan torpedo signal that specifies dorsalized follicle cells initiate a cascade of gene activities that create the dorsal ventral axis of the embryo. The activated torpedo receptor on the presumptive dorsal follicle cells inhibit the expression of the pipe gene. On the presumptive ventral end, in the absence of Gurkhan protein, inactivated torpedo receptor facilitates the expression of pipe gene. Pipe protein modifies an unknown protein X and allows it to be secreted from the ventral follicle cells. Pipe activates the noodle protein, which is secreted to the cell membranes of the neighboring ventral embryonic cells. Noodle protein interacts with its modified factors to split the product of the gastrulation defective genes. The GD protease cleaves the snake protein, activating snake protease, which in turn cleaves the Easter protein. The cleavage of Easter protein by snake protease activates the Easter protease that cleave the spatzel protein. The gene product of gastrulation defective snake and Easter are the serine proteases that are activated when they are cleaved and limited to the most ventral portion of the embryo. When the cleaved spatzel portion is now able to bind to its toll receptor found in the oocyte cell membrane, and only the ventral cell receives the toll signal. The signal activates a protein kinase called a PEL and other protein tubes necessary for binding PEL to cell membrane. The activated PEL kinase can phosphorylate cactus, causes cactus protein degradation and separate the cactus protein from the dorsal protein, allowing dorsal to be translocated into the nucleus and ventralize the cells. Initially, the mRNA transcript of the mother dorsal gene is placed into oocyte by the nurse cells. Therefore, dorsal protein is found throughout the syncytial blastoderm, but after fertilization, the dorsal protein is only translocated in the nuclei of the ventral follicle cells of the embryo. In the nucleus, dorsal protein acts as a TF, binding certain genes to the activate or repress their transcription. Dorsal protein is found in the nuclei of every ventral follicle cell. Reciprocal interactions between the oocyte and egg chamber specify the dorsal ventral polarity of the embryo. Specification of cell heat by the gradient of dorsal protein. Dorsal aminosolution, dorsal ectoderm, lateral endoderm, ectoderm, neurogenic ectoderm, mesoderm. Dorsal protein activates the zygote genes rhomboid twist FGF8 receptor and snail depending on its nuclear concentration. Twist snail TGF8 receptor gene is present from mesoderm. Only rhomboid gene represent from the neurogenic ectoderm. Action of dorsal protein in ventral cells. Dorsal protein inhibits genes that give rise to the dorsal structure. Then, but dorsal protein activates ventral-specific snail gene whose product inhibit the transcription of rhomboid and prevent ectoderm formation and provide mesoderm specification fate to the ventral cells of the embryo. Segmentation genes. The transition from specification to determination in Drosophila is mediated by segmentation genes that divide the early embryo into repeating series of segmental primordia along the anterior-posterior axis. The gap genes. The gap genes are activated or repressed by the maternal effect genes and are expressed in one or two broad domains along the anterior-posterior axis. The hunchback gradient is particularly important in establishing the initial gap genes expression pattern. By the end of the nuclear division cycle 12, hunchback is found at high levels across the anterior part of the embryo and then forms a steep gradient through about 40 nuclei near the middle of the embryo. The last third of the embryo has undetachable hunchback level. The transcription pattern of the anterior gap genes are initiated by the different concentration of hunchback and bicoid proteins. High level of hunchback also prevents the transcription of the posterior gap genes. Knirps and giant. The gradient of the caudal protein, highest at the posterior pole, is responsible for activating the abdominal gap genes and Knirps and giant at the embryo. After the initial gap genes expression pattern have been established by the maternal effect gradient and hunchback, and they are stabilized and maintained by the repressive interactions between the different gap genes predict themselves. Gene expression experiment shows that giant and Krüppel was strong mutual repressor, as are hunchback and Knirps. For example, if hunchback activity is lacking, the posterior domain of Knirps expands towards the anterior. The end result of this repressive interaction is the creation of a precise system of overlapping gap mRNA expression patterns. High levels of bicoid and hunchback induce the expression of giant, while Krüppel transcript appears over the region where hunchback begins to decline. There is a strong mutual inhibition between hunchback and Knirps and a strong mutual inhibition between giant and Krüppel. And the pair rule genes. The first indication of segmentation in fly embryo comes when the pair ruled genes are expressed during cell division cycle 13 as cells begin to form at the periphery of the embryo. The transcription pattern of these genes divide the embryo into regions that are precursors of the segmental body plan. One vertical band of nuclei express a pair rule gene, the next band of nuclei does not express it, and then the next band expresses it again. The result is a zebra stripe pattern along the anterior-posterior axis dividing the embryo into 15 subunits. 8 genes are currently known. They overlap one another so as to give each cell in the para segment a specific set of transcription factors. The primary paired rule genes include hairy, even, skipped, and errant, each of which expressed in 7 stripes. Each skipped enhancer is composed of modular units arranged such that regulate a separate stripes or a pair of stripes. The DNA regions of pair-ruled genes contain binding sites for gap proteins that determine whether or not a pair-ruled gene is transcribed. The border is maintained by repressive influence from giant, while posterior border is maintained by Krüppel. Once initiated by gap gene proteins, the transcription pattern of the primary pair-ruled genes become stabilized by interactions among their products. Early in division cycle 14, FTZ, Frusi Terasu mRNA and its protein are seen throughout the segmented portion of the embryo. However, as the protein from the primary pair-ruled genes begin to interact with FTZ enhancer, the FTZ gene is repressed in certain bands of nuclei to create interstripe regions. But FTZ proteins interact with its own promoter to stimulate more transcription of FTZ where it is already present. Pair-ruled gene, hairy, even, skipped, red. Secondary pair-ruled gene, FTZ, OPA, ODD, slopy paired, SLP, and paired. And the segment polarity genes. The interactions within the cell is mediated by the segment polarity genes. They accomplish two important tasks. They reinforce the parasegmental periodicity established by early transcription factors. Through the cell-to-cell interaction, cell fates are established within each parasegment. The normal development indicator only one row of cells in each parasegment is permitted to express the hedgehog protein, and only one row of cells in each parasegment is permitted to express the wingless protein. The engrailed gene is activated in cells that have high levels of the even skipped fusi-tarazu, and the wingless gene is transcribed when neither E or FTZ genes are active. The types of engrailed transcription mark the anterior compartment of each parasegment, and the wingless transcription mark the posterior compartment of each parasegment. The continued expression of WG and EN is maintained by reciprocal interaction between the engrailed and wingless expression cells. Wingless protein is secreted and diffused to the surrounding cells. In those cells that are competent to express engrailed wingless protein is bound by the fragile receptor, which enables the activation of the EN gene via the WNT signal transduction pathway. Armadillo is the Drosophila name for beta-catenin. Engrailed protein activates the transcription of hedgehog gene and also activates its own EN gene transcription. Hedgehog protein diffuses from these cells to bind to the patched receptor on neighboring cells. This binding prevents the patched protein from inhibiting signaling by the smoothened protein. In this way, the transcription pattern of these two types of cells is stabilized. This creates a subtle boundary as well as signaling center from which hedgehog and wingless protein diffuses across the parasegment. The diffusion of these proteins is thought to provide the gradients by which the cells of the parasegment acquire their identities. Then next, the homeotic selector genes. After the parasegmental boundaries are set, the pair rule genes and gap genes interact to regulate the homeotic selector genes, which specify the characteristic structure of each segment. Two regions of Drosophila chromosome 3 contain most of the homeotic genes. The first region is known as the antennapedia complex, contain the homeotic genes labial and antennapedia. Then sex comb reduced, deformed, and proboscipedia. The labial and deformed genes specify the head segment, while sex comb reduced and antennapedia contribute to giving the throat segments of their identities. The proboscipedia gene appears to cut only in adults, but in its absence, the labial pulps of the mouth are transformed into legs. The second region of the homeotic gene is the bithorax complex. The genes found in this complex are ultrabithorax, which is required for the identity of the third thoracic segments, abdominal A and abdominal B, that are responsible for the segmental identities of the abdominal segments. The chromosome that contain both this complex is called as homeotic complex. Bithorax complex, ultrathorax, required for the identity of the third thoracic segment, abdominal and abdominal B, responsible for the segmental identities in the abdominal segments. In homeotic mutants, the specific identities of the segments are changed. When the ultrabithorax gene is deleted, the third thoracic segment characterized by halters is transformed into another second thoracic segment. The result is a fly with 4 wings. Similarly, antennapedia protein usually specifies the second thoracic segment of a fly, but when flies have a complete dominant mutation where the antennapedia gene is expressed in the head as well as thorax, legs rather than antennae grow out of the head sockets. In the head region, antennapedia protein represses the enhancers of homeothorax and eyeless genes, which encode transcription factors that are crucial for antennae and eye formation. In the recessive mutation of antennapedia gene, it fails to be expressed in the second thoracic segment and antennae sprout into leg position. Initiating and maintaining the pattern of homeotic gene expression. The initial domain of the homeotic gene expression are influenced by the gap genes and pair rule genes. For example, the expression of ABDA and ABDB genes are repressed by gap gene proteins, Hunchback and Kruppel. This inhibition prevents these abdominal specifying genes from being expressed in the head and thorax. Antennapedia is originally transcribed in the parasegment 4 specifying mesothorax segment. ANTP expression is negatively regulated by all the homeotic gene products expressed posterior to it. In other words, each of the bithorax complex genes represses the expression of ANTP. If the UBX gene is deleted, ANTP activity is extended through the region that would normally have expressed UBX. If the entire bithorax complex is deleted, ANTP expression extends toward the abdomen. Such larvae does not survive.